Hello friends, how are you doing? And I hope you really all are having a great and fun time. Okay, so we are doing the chapter Neural Control and Coordination, and we will talk about some of the CBSE pattern question exercise regarding this particular chapter. What is the function of the eustachian tube? Now these eustachian tubes, these are actually present between the middle ear and the nasopharynx. And what does the nasopharynx? We know that the pharynx is a chamber that is situated behind nasopharynx, is that chamber which is situated behind the nose and the nasal cavity. Okay, so the nasopharynx is connected to the middle ear by means of this eustachian tube, and this eustachian tubes makes it eustachian tubes makes it necessary, or makes sure that the air pressure on both the sides of the middle ear are well balanced or the same. So I am going to write this down, right, that the eustachian tube connects the middle ear, right? It connects the middle ear to nasopharynx, and we have already said what is the nasopharynx. That is that part of the pharynx which is behind the nasal chamber and the nasal cavities. That is that part of the nasal cavity, okay, that is that part of the pharynx, we can say, of the pharynx which is behind the nasal cavity. And nasal chamber, right? The eustachian tube connects the middle ear to the nasopharynx. That is that part of the pharynx which is behind the nasal cavity and the nasal chamber, Hence, the eustachian tube equalizes the air pressure, okay. Hence the eustachian tube equalizes the air pressure on either side of the middle ear. So this is the function of the eustachian tube. The eustachian tube connects the middle ear to the nasopharynx, that is that part of the pharynx which is behind the nasal cavity and the nasal chamber. Hence the eustachian tube equalizes the air pressure on either side of the middle ear. This is a very important, you should know what is the eustachian tube and what does it connect and what is its function, right. With this, we move on to the next question. What are the neurotransmitters? Now the word itself tells you a lot, that these are something related to transmission, right? These are something related to transmission and involved in the nervous tissue, right? And you remember this is a 1 mark question. So the neurotransmitters the neurotransmitters are chemical messengers, you can say are chemical messengers, right. The neurotransmitters are chemical messengers that are involved in the transmission of nerve impulse, that are involved in the transmission of the nerve impulse, right. The neurotransmitters are chemical messengers that are involved in the transmission of the nerve impulse, and the best example for this will be acetylcholine, right. Example, acetylcholine, which is generally called ACH in the abbreviated form. Now, what we see, these acetylcholine, when you are talking about the junctional, synapse, synapse, and in the pre-synapse membrane, okay, when in the when in the pre-neuron, what we will see that these acetylcholine or the chemical messengers, they are within the synaptic vesicles, right. What happens, there is an influx of calcium and these are being released from the vesicles, these acetylcholine, which will take the the message, okay. They are basically for the, because the impulses are traveling from one neuron to the other, and whether whether it is going to the motor neuron or to the brain, that depends on what what direction it is going. So, these will these acetylcholine, which are the chemical messengers, these will take that particular information across the synaptic cleft and then pass it on to the postsynaptic membrane. So, there is a whole thing, we have discussed about this in the chapter. The neurotransmitters are chemical messengers that are involved in transmission of the nerve impulse. Example is acetylcholine. We move on to the next question. Name the band of nerve fibers that joins the two cerebral hemispheres, right. It is very, it's just one word, the band of nerve fibers, the band of nerve fibers that connects the two cerebral hemispheres, is the corpus callosum. The band of nerve fibers that connects the two cerebral hemispheres is the corpus callosum, right. So, these are the band of nerve fibers that is connecting the two cerebral hemispheres. We move on to the next. Where is the cerebrospinal fluid present in a body, and mention any two of its functions, right. As the question has moved to 2 marks, so they are mainly asking the two sub parts of the question, that where is CSF present in our body. The cerebrospinal fluid, the CSF, which is nothing but the cerebrospinal fluid, is present, right, in the brain and the spinal cord, in the brain, right, and spinal cord, right. Now, this is a body fluid which is being secreted by the ependymal cells. Okay, this is a body fluid. This is a body fluid present in the brain and spinal cord, secreted by the ependymal cells. Body fluid secreted by the ependymal cells. Okay. So, this is a body fluid which is secreted by the ependymal cells. The cerebrospinal fluid is present in the brain and spinal cord. This is a body fluid being secreted by the ependymal cells, and the functions. What are the functions of CSF, cerebrospinal fluid? First of all, since this is a fluid present in the brain and spinal cord, so basically, it will protect the brain and spinal cord against the trauma. This will protect the brain and spinal cord. Okay. This will protect the brain and spinal cord against the trauma. Right. Secondly, what we will see? Secondly, what we will see that the CSF, apart from protecting the brain and spinal cord, this provides nourishment to the nerve cells. Okay. This will also act as nutritive tissue, right. So, we have mentioned two of its function, that it will also act as the nutritive function, nutritive tissue. So, the cerebrospinal fluid is present in the brain and spinal cord, right. It is a body fluid which is secreted by the ependymal cells, and this will protect the brain and the spinal cord against the trauma, right, against the trauma, and this will also act as the nutritive tissue. Friends, you note down this, and then we will continue after just a short break. Right, friends, so I hope you must have noted down, right. And now we will be talking about the next question. So, where are the cerebrospinal fluid present? These cerebrospinal fluid are present in the brain and spinal cord, and they will protect, they will be protecting the brain and spinal cord, that is a nervous tissue, and apart from that they also serve as the nutrition secreted by the ependymal cells. Let us move to the next question. How is the shape of the cornea maintained? We all know that the eye has two liquids, and before that, where are these two liquids situated? Yes, there are two chambers in the eye. One is the aqueous chamber and one is the vitreous chamber. The aqueous chamber having aqueous humor and the vitreous chamber having vitreous humor absolutely are an eye, a body. They cannot be a more better machine than our body and various organs of the body. So, what we see when we try to draw, right, the eye, somewhat like this, right. And then what we see, that over here will be the lens which is suspended by the various structures like the ciliary body, the suspensory ligaments and all that. Right, and then here we see this part is bulging out forming the cornea, and then the choroid, the sclera, the choroid and the retina will be present all over the body. Right, the sclera, the choroid and the retina, and here so we see that this is the portion which is called the aqueous chamber, which is filled with the aqueous humor. This is the lens and this is the vitreous chamber having the fluid called vitreous humor. Right. So, this is the vitreous chamber having the fluid called the vitreous humor, and this is aqueous chamber having aqueous humor. Right, this is a lens, and hence we see that this is the cornea, and thus the cornea we can say the cornea's shape is maintained by this fluid called the aqueous humor, aqueous chamber having the aqueous humor. So, the cornea shape is maintained by the aqueous humor, right. The aqueous humor also will provide nutrition to the cornea. The aqueous humor will also provide nutrition to the cornea. The cornea's shape is maintained by the aqueous humor. The aqueous humor will also provide nutrition to the cornea. We will move on with the next question. What type of striated muscles constitute the muscles of the eyeball, and how the specific type of muscle advantageous? Right. So, 6 types of striated muscles, right. 6 types of striated muscles constitute what is what we call as the muscles of the eyeball. Striated muscles constitute the striated muscles of the eyeball, right. The inferior and superior oblique muscles, right, the inferior and superior oblique muscles. After this we have the lateral and medial rectus, lateral and medial rectus muscles, and then inferior and superior rectus muscles, right, the inferior and superior rectus muscles, inferior and superior rectus muscles. So, these 6 muscles which constitute the striated muscle, that is, yes, we can, they are in our control. For example, if you want to move the lateral and medial rectus, the medial will move the eyeball, okay, towards the medial side, right, towards the medial side, towards the inner canthus of the eye. And what will be the lateral rectus doing? They will be moving the eyeball away from the inner canthus of the eye, that is moving towards the outer canthus. So inferior and superior oblique, the lateral and medial rectus, and the inferior and the superior rectus muscles. We now move on to the next question. Why does a nerve impulse flow more rapidly in a myelinated nerve fiber than in a non-myelinated nerve fiber? So what we see, in, say, this is a non-myelinated nerve fiber, right? This is the whole length of the, the nerve fiber. What is it? We all know that when a nerve impulse is flowing, impulses going or propagating, how does it go? It gets propagated by means of the nerve impulse. The nerve impulse is getting propagated from one point to the other, from the second to the third to the fourth, and so on. But here, right, that is how. However, the action potential. So the nerve impulse is flowing, by the generation of action potential. So say the action potential is generated here, here, here. So the nerve impulse comes here, here, here. But, okay. Say, there is a myelin sheath. There is a myelin sheath here, and the myelin sheath are absent at places called the nodes of the Ranvier. Nodes of the Ranvier. So what happens? That action potential is generated here. It will be generated here. It will be generated here. I mean, at the next node of Ranvier. So, in case of a myelinated nerve fiber, we will see that the impulse is generated, the action potential is generated at the nodes of Ranvier, where the myelinated nerve fiber myelin sheath is absent, and hence the impulse jumps. Okay, so we will write this. In myelinated nerve fibers, action potential, right, the action potential jumps from one node of Ranvier, to another. Hence, the impulse action potential jumps from one node of Ranvier, and the impulse takes less time. Okay, node of Ranvier, this is a place where myelin sheath will be absent, where myelin sheath will be absent. Okay. Now we go on to the next question. What do you understand by the binocular vision? What do you understand by the binocular vision? Binocular vision is seen in mammals. This this is a distinctive ability where the two, having the two eyes and the two eyeballs can focus, okay, on one object at a single time. That is, a three-dimensional object can be seen at once. It is absent in infants. Okay, so binocular vision as seen in mammals. Right. Binocular vision as seen in mammals is the ability to focus an object as a single object by means of two eyeballs, that is, both the eyeballs or both the eyes are bringing, are focusing that point of object, and hence that object, three-dimensional object, can be seen as one object only by the help of two eyes or two eyeballs. So that is what is a binocular vision. You can just elaborate on this more since I have explained you. Right. We move on to the next question. What is night blindness? What causes night blindness, and how can it be remedied? Right. Night blindness. See, night blindness is basically a disease where you cannot see in, where your dark vision is impaired. Okay, here the dark vision is impaired. Okay, the dark vision is impaired. Right. Now, the rods are the photosensitive pigments which are responsible for seeing in the dark. Okay, the rods are the photosensitive pigments, which are responsible to see in the dark, which are responsible for dark vision, right, which are responsible for the dark vision. So what we see, deficiency, and these rods are made up of the protein called the rhodopsin. So what we see, deficiency of vitamin A, causes night blindness in children, and xerophthalmia in adults. So what happens, if in children, and xerophthalmia in adults, if in children the cod liver oil, the leafy vegetables, those which are rich in vitamin A are not given to children, then that rhodopsin protein will not be synthesized, and hence the rods will be less in them. How can it be remedied? Yes, give more of vitamin A, try to give more of vitamin A in such children, when they are in the developing stage, right. We go on to the final question. How do rods and cones of the human eye differ from each other chemically and functionally? Yes, absolutely. The rods and the cones. Now, as I said, let us see the difference each other chemically. They are composed of the protein rhodopsin, and these are composed of the protein iodopsin, right. The rods are responsible, right, the rods are responsible for night vision, that is dark vision, and the cones are responsible for color vision. That is, we will see that in the absence of the cones, the color blindness will be seen. Okay, those color blindness will be later on can be checked by the Ishihara cards and all that, right. So they are responsible for daylight vision, and the color vision and the color vision. That is, that means one thing, that these rods are active only, when low intensity of light, and the cones are active when there is a high intensity of light, right. So, active when there is a low intensity of light, and this cones are active when there is a high intensity of light. And this is one of the differences, some of the differences, both structurally and as well as functionally, between the rods and the cones. So, friends, we have come to the end of this questions exercise. Get back, revise this. Any doubts, you can always get back to me. Thank you.